Afternoon, class team. Thank you for joining into our new topic of today, transcription, Great Hope Life Sciences. It is one of the key areas that most of the students they have problems, and sometimes it's confusing because of some scientific expressions that are in there and terminologies and stuff. So right now, I just don't want to waste your time. I'm going to be presenting to you transcription process, the process through which mRNA is formed from DNA. On the board right now is just a structure or the diagrammatic representation of the process of transcription. What we are seeing here is that this is a DNA structure that is being displayed from the nucleus of a cell. So what happens is that mRNA is made from DNA. What I need you to understand is that this diagram, it sometimes comes in the exam, and then you are not able to give the correct labels of it. So let me just begin by doing the labeling. So what is happening here is that this diagram is representing, or we are talking about the transcription processing. The transcription process here, I'm just underlining the topic, transcription process. So what is happening here is that you see there is a membrane that is shown here. This membrane is called the nuclear membrane. That is labeled here is the nuclear membrane, which we are seeing shown by these broken, or by these black marks that are surrounding the diagram, or which are surrounding the DNA structure there. So this whole diagram, or these all points, are marking the nuclear membrane. The nuclear membrane has got gaps between it, which I can label to say these gaps here, for example, where we are given the written the nuclear pore. The nuclear pore, there are gaps that are found on the nuclear membrane, which actually enables the mRNA to move out of the nucleus after it has been formed. So again, I'm sure you can see this is the DNA structure. It is the DNA. It's a DNA molecule that you are seeing there. And then obviously here it is already labeled, that is mRNA, which is labeled here. The mRNA is labeled there. So what is happening here is that this is a process from which mRNA is formed in order to prepare for what? For protein synthesis. That is again a stage at which the DNA segment is being copied from what is being copied to mRNA. So this diagram, it comes like that, and please take note of it, and don't make any mistakes. I'm going to quickly go to the next step. So don't confuse transcription to that other stage. What is it to the previous process of DNA replication? This is not replication, because we are not seeing a formation of a double strand. We are only seeing a formation of a single strand that is being formed from a certain template from one strand of the DNA that is used as a template. Hence, we can be able to explain and say that confidently we can confirm that that is transcription. So what is transcription? It is actually a process. Transcription is a process of copying. It's a process of copying a segment of DNA into RNA for the purpose of gene expression. For the purpose of gene expression, when a segment of DNA is copied into RNA for purposes of gene expression, that is what you call transcription. So some segments of DNA are transcribed into RNA molecules, are copied, are transcribed into RNA molecules that can encode for protein called mRNA or messenger RNA, which you can see sometimes written as mRNA. which stands for messenger RNA. When messenger RNA is formed, the process responsible for that formation is called what? It is called transcription. Transcription is when mRNA is formed. Messenger RNA is formed. When it is formed, we call the process transcription. So RNA polymerase an enzyme. It is an enzyme. This is an enzyme that actually, it catalyzes an enzyme, it activates, it speeds the process. It binds to a specific sequence on the DNA called the promoter, located at the beginning of a gene. So the polymerase enzyme, it actually speed up the process of what of the formation of mRNA by binding to a specific sequence of the DNA, which we are saying the promoter that is located at the beginning of the gene. This binding unwinds the gene. The binding, it unwinds the DNA segments. It unwinds the DNA segment in mRNA, and the RNA polymerase reads the DNA template strand to synthesize. So what is happening is that it reads the what? It reads the DNA template strand to synthesize or to form, where it is supposed to form the what? The RNA. So unlike in the DNA, the RNA nucleotide uracil is used instead of thymine. I'm sure this I've explained it enough. I'm not gonna quick go back to talk about it, where I said that when we are in RNA, it is A is equals to U, meaning to say A and U, they pair up together. A and U, they pair up together, unlike in DNA. So I don't think I have to go back and explain anything about this. I've explained when we look at the comparisons between the structures of DNA and RNA. So once the mRNA molecule is synthesized that when it is formed, after it has been formed, when it is formed, it undergoes process where non-coding regions are removed. The non-coding, we are saying there are various types of proteins. Certain proteins are non-coding, so when they are identified, they are removed, and a cap and tail are added to stabilize to stabilize the mRNA before it exits the nucleus. Before it exits the nucleus. So what I want you to understand is that the process of the process of formation of mRNA, it is called transcription, where a small piece of DNA, it unwinds, and then the weak hydrogen bonds breaks, making one of the strands of the DNA being used as a template to form and copy a single strand that we call the mRNA. So what I want you to look at, or what I'm going to quickly just add onto this one, is that we have got that diagram which I showed you there, the diagram which I was showing, which you saw at the beginning there. So we are saying a small piece of DNA, it unwinds. When it unwinds, what happens is that the weak hydrogen bonds that are holding the complementary bases, they break. So we have got our DNA, we have got our nitrogen bases there. So the nitrogen bases, we have got the weak hydrogen bonds there, so they break. So we are going to see from the free-floating nucleotide bases that are within the nucleus, a single strand is going to be formed. A single strand is going to be formed there. So we call this single strand, I remember it is formed by what? By it is formed by phosphate sugar molecules, which is the ribose and phosphate molecules are forming the single strand. So we have got the single strand there, which we are saying it is formed. When it is formed, we have got the nitrogen bases, which are controlled by one of these ones, which is, we are saying this is the template or the original strand, which we can give, for example, that we have got A, U, C, G, U, as to say these are the nitrogen bases that are found on the on the DNA molecule. Sorry, we don't have U there. We are saying on the DNA molecule. Obviously, we are saying this is a DNA molecule. Don't forget that we have got what? We have got T. I'm saying this is a DNA molecule, hence it has what? It has got T. So what we are saying is, according to the order of the nitrogen bases that are found on the what? On the one of the strands of DNA, this is going to define the order of the nitrogen bases that are going to form on the single strand that we have said that it has formed here. So according to the pairing that we said, A, it pairs with what? It pairs with the U. So we are going to have our U here, and then T, it pairs with what? With A. We have got C here, it pairs with what? C pairs with what? With G. G pairs with what? With C, and then T, it pairs with what? With A. So these are nitrogen bases that are written in blue are the nitrogen bases that are going to be, or that are found on the what? On the single strand, this one. On the single strand, which we are saying it is what? mRNA. It is mRNA that is formed there, the single strand. So when it is formed, when this single strand is formed, it moves out of the nucleus. It moves out of the nucleus, and then it has to rest as a single strand, which it lies below there as a single strand. And then we have got this existing or of appearing there, where I'm saying it is the same, it is the same, it is the same, it is the same molecule which is moving from, let's say, 1, it is coming to 2. So 2 we have got, I mean, it is the same thing, which is this the mRNA molecule. So we have got our U, we have got our A, we have got our G, we have got our C, we have our A. These bases are just the bases that are coming from the single strand that was formed there. So in that respect, we are saying we have got our what? Our mRNA strand that is formed. So in a nutshell, understanding that we have got the nitrogen bases that are existing, we have got the nitrogen bases that are there. Don't forget that in between nitrogen bases, we have got what? We have got the weak hydrogen bonds that support the corresponding or the complementary bases that are found on the strand of what? On the complementary bases which are existing on this DNA molecule. So what we need to understand is that DNA unwinds, so therefore it becomes a single strand. As I can see, I can maybe break it from this position. I can break it to say this is a small piece of DNA that unwinds, which I'm saying this piece, it did unwind, and then as it unwinds, the weak hydrogen bonds which were holding the complementary bases from the both complementary bases, I'm talking about the bases that are found on both strands of this DNA molecule. So the DNA molecule which we are talking about here is that we are saying this the strand. I can say maybe I can say strand 1, and then I will say strand 2. So the strand 1 and 2 are representing the original strands of the what? Of the DNA structure. So the DNA structure, it is actually having its own basic, it has got its 2 strands that are original. So one of them, in this case, we are saying strand one was used as a what? As a template to form the strand 3 that I can maybe mention here, or I can label to say strand 3 here. Strand 3 was formed from the DNA structure, which we are saying is the first stage of copying of a gene, where it is copied to form what you call the mRNA. So what is happening is that structure 3 is formed as a result of the template, which is template 1, that has been used as a what? As a reference or as a guideline information of strand 3. So strand 3, as you can see it there. So what happens is that the two uh, uh, I had said that forming the one strand acts as a template there, which I'm saying one strand number one, it is used as a template in this regard. So it is used as a template to form a complementary strand with which is mRNA. So mRNA is strand number 3. It is formed as a result of a complement, or it is complementing to one of the strands, which is strand number 1 there. So in that respect, uh, using the free nucleotides from the what? From the nucleoplasm. So the nucleoplasm, don't forget, like I said in our diagram there, that we have got our DNA structure here, which is within the what? The nucleus. So the nucleus has got free floating. I can maybe give to say these things that I'm putting the dots here is the nucleoplasm that is being referred to. It is the nuclear plasm. So the nuclear plasm, it is having all the structures, or it has got the nucleotides, which are the building blocks of the DNA. So the nuclear plasm, it has got the phosphate, it has got the sugar, it has got the nitrogen bases. So having said that, don't forget that both nucleic acids are formed from what? From nucleotides as the what? Is the building blocks. So what is happening is that strand which is newly formed that we are seeing here, the newly formed strand, it is formed from the nucleoplasm that we are talking about. To say this newly formed strand, the newly formed strand class, it's formed from the nucleoplasm, and then the process that I'm talking about is therefore that we call the what we call the transcription, and then three adjacent bases on mRNA makes a codon which codes for an amino acid. So adjacent, we are talking about to say on the mRNA that is formed, they code for a codon, or they make up a codon which code for an amino acid that is to be what? That is to be formed. So if I'm going to demonstrate this on the next diagram that I've used here, what is happening is that three of the adjacent, we are saying three adjacent bases of the mRNA. I said this is mRNA class. This is mRNA. So we are talking about these three of them, like to say from here to there, they are forming what you call a codon. These are codon. We are saying they form a codon, which we are saying it is actually the three nucleotide bases that are found on the mRNA, which codes for a what? For an amino acid, which we are going to look at as we go to describe the next step translation. So that is the codons there. Codons are found on mRNA, a single strand that is formed during process of transcription using one of the strands of DNA as a template. Then that is what? Transcription. So I want us to understand to say that when the process is completed, I said when it lies like this as a single strand here, as where it is lying, it means there is a movement of the mRNA out of the nucleus. How does it move out of the nucleus? I told you that there is a nuclear pore, which I defined and I labeled in the first diagram there to say the gaps that you are seeing here, these are gaps. These gaps, we call them nuclear pores. These are called nuclear pores, these ones. They are called nuclear pores, which we are saying they are gaps through which mRNA will escape from the nucleus to come and lie itself or align itself in the what? In the cytoplasm. So it moves out of the nucleus. mRNA moves out of the nucleus after it has been fully formed. So therefore, the DNA, it reforms, or it retakes the same strand, the structure of its original structure of a double helix after the mRNA is formed. So let's go and look into this question of saying what happens when it moves out of the nucleus. So movement of mRNA out of the nucleus, I said it moves through the nuclear pore, movement of mRNA out of nucleus. Remember, it is formed inside the nucleus, and don't forget that DNA that we are talking about is found in the nucleus. So after it is formed, it moves out of the nucleus. It actually through the it moves out of the nucleus. One, we are saying through nuclear pore. I showed you the gaps that are there, the openings that are surrounding the nucleus there. And then what happens is that when it moves out of the nucleus through, it moves out of the nucleus through the nuclear pore into the cytoplasm. It is moving into the cytoplasm, and then where it attaches itself to the what? When it moves into the cytoplasm, it attaches itself. It attaches itself when it attaches itself where? To the ribosome. I defined last time to say that is the manufacturing site for proteins, the ribosome. It attaches itself to the ribosome, so therefore it prepares itself for the process of what? For the process of transcription, translation, protein synthesis. So movement of mRNA out of the nucleus, it happens through the nuclear pore into the cytoplasm, where it attaches itself to the ribosome. So having said that, that is exactly the entire process that you can define and explain, the process of what? The process of transcription. Process of transcription, you can explain it in that regard. You can explain it in that respect. And then if you do that, you are making sure that your points and your marks that you should get on this question, they are fully catered for. They are fully marked, and you won't lose any marks there. You won't lose any marks. Copying of a segment of DNA into RNA for purpose of gene expression. Remember that proteins are used for, we have got two forms of cell division, the mitotic cell division and mitosis, and mitotic and meiosis cell division. So what is happening is that this process of copying of a DNA into RNA for purpose of gene expression, it is a process that is, or it is happening to prepare for the process of what? Of mitotic cell division, which is the growth and the replacement of dead cells. So dead cells, when they need to be, or when they are to be replaced, a gene or the genetic code must be found or must be readily available into the new cells that are to be replacing, or that are replacing the dead cells, as well as growth. So that is what is happening with regard to the process of what? Of transcription. So I also said that you need to know that this diagram is representing transcription process. This is a transcription process which sometimes you are going to see there, and you will be asked to label certain things there, certain areas or certain components. So please don't forget that in this case, you might be given that the molecule, the DNA nitrogen bases are given there, and then you won't be given those of what? Of the mRNA that is being formed. So it is your duty, and it is your responsibility to ensure that you don't forget that when it is mRNA that is formed on itself, we are no longer having T, but we are having U, which is the reason why you see that we are having this U there. We have got also that U here. It is actually identifying to show you that this is an mRNA, or it's an RNA molecule that is being formed. So remember to say, what is the essence of the DNA? You see, on this case, if they are going to come and ask you something to say template strand of DNA, explain the significance or what is the function of the template strand of DNA, it is actually to decide, to define and give a blueprint towards the formation of the mRNA, defined by the order of the nitrogen bases that are what? On the template strand of the DNA. So I'm sure we have done and dwelled enough on this. Thank you very much for your time and all the best. Let's keep on following and subscribing and also sharing to friends and family. We are going to talk about translation tomorrow, which is now what is the significance or what is the function of mRNA. The way mRNA is performing its function is translation or protein synthesis. So just follow to the next presentation where we are going to talk about what translation, and then we'll talk about DNA profiling. We'll talk about the every other aspect of DNA, and we complete the unity. So therefore, we can go to the next topics. But anyway, we are together here until November, December. So therefore, our distinctions are guaranteed. Don't forget to subscribe and share, and always know that Metric Hub is there for you. Let's share Metric Hub to friends and family. So therefore, all of us, we can make it. You have a beautiful day. Thank you.